Okay, good afternoon. This is part two of the experiment. And in this case, we're going to discover fruit browning or enzymatic browning in fruits. So these are like materials. I use milk, I use water, an apple, two bananas, lemon juice, tape, something to boil, water, marker, vinegar, and some plates, and an apple cutter. The first thing that I did was cut the apple into different slices. Then to one slice, I poked it and I placed it in a plate which marked poked. Then I had control, which was just the cut up slice. Nothing had done to, I had done nothing to it. Then I let them rest for 35 minutes, and this is what I got. So as you can see, and I noticed that the one that I poked was a little bit browner. Then I did the same thing, but this time I I used 5 samples. For the 4 of them, I added different solutions, and I had one control which I added nothing to. So for the first one, I added vinegar. To the next one, I added milk. I added lime to the next one, and then I added water to the last one. And I poked all of them. This is me putting the solutions on top of each of them and poking them. I let them sit for 30 minutes, and this is what I got. As you can see, 4 of them, the control, the water, the vinegar, and the milk had more browning than the lime one. The lime one relatively stayed the same color, or it had some browning, but just a very tiny bit. Not very much. I could not even see a little bit of browning, but the other 4 did have browning. The other thing that I did was I dipped a piece of a banana sample in boiling water, and then I observed it for 30 minutes. And after for 30 minutes, sorry, for 30 seconds. And when I took it out, I observed it, and this is what happened. I noticed that the tip that I boiled had browning. It became brown. Then I peeled it, and the inside remained intact. And the reason for this is because only the outer layer of the banana was damaged and not the inside, so the inside remained the same. Then I took a banana, I tipped it, however, whichever design I wanted, and I put it in the refrigerator for 1 week. And this is what it looked like after 1 week. As you can see, the part that I didn't tip became brown, and the part that I did tip remained yellow. So now let's explain what happened in my results, in my interpretation of the results. So the discoloration of the fruit is due to the fact of an enzyme called polyphenol oxidase, which oxidizes phenolic compounds such as 2,4-dihydroxyphenol, which is found in the cells of the fruits, in the surface of the fruits. And so the oxidation of this phenol results in the formation of melanin, which is a pigment, the brown pigmentation which happens after the cells of the fruit is damaged. For that reason, in the first experiment in which we had only the poking and the one that I didn't poke, we noticed that one became browner, a lot browner than the first one. And this is because I damaged more cells, and then hence more PPO, or the polyphenol oxidase, was released. And as polyphenol oxidase is released, it oxidizes the phenols, but it has to occur in the presence of oxygen. That's why it's an oxidation reduction reaction. Oxygen must be present in order for it to occur. And also, in these other 5 samples that I used, we noticed that the one with the lime did not change at all. And this is because the polyphenol oxidase, which is the enzyme, does not function very well under acidic conditions. And as such, it was not able to oxidize the phenol. That's why there was no formation of the melanin color. In contrast, the other 4, which were in basic conditions, they had no there was no effect on them. The browning occurred as it normally would. And then in, in this case, in the banana, when we dipped it, we damaged the cells. That's why polyphenol oxidase was released from the cells as they were damaged, and then as it reacted with oxygen, um, the formation of the melanin compound was formed as dihydroxy 2,4-dihydroxyphenol was oxidized. And for the banana that we left in the fridge for 1 week, the reason for why this happened, the reason why only a section of it remained intact and the other became brown is because we know that bananas are a type of food that thrive in warm weathers, and as such as by putting them in the refrigerator, we expose them to a temperature which they are not accustomed to, and so the cells are damaged. And remember that we said that when the cells are damaged, they release the PPO from their cells and react with oxygen, and hence the melanin is formed, the melanin pigment. But when we taped a part of the food, we prevented it from being exposed to oxygen. And as we said before, this reaction or this oxidation reaction requires oxygen in order to occur. And so in the absence of oxygen, the polyphenol oxidase is not able to reduce the 2,4-dihydroxyphenol to melanin. And that is why a part of the banana remained yellow and the other became brown after 1 week. So that's my conclusion for this part of the lab. Thank you.